afternoon on this beautiful sunny day. Um, I want to talk a little bit about our pathway going from what we were using before, and I'll get into that, to the fusion that we're using today. Whoops. Make sure I'm hitting the right one. Which one? Right here? Oops. Sorry about that. There, right there.
You just tap on this, and then yes.
Oops, how do you go back? There we go. All right. For disclosure, Hologic did pay for my travel expenses to CVS. Oops, we went backwards. Hang on, we'll get it. There, I got it. I got it. So I wanted to tell a little bit about University of Vermont Medical Center for those who may not know about us up in northern Vermont. We are in Burlington, Vermont. We're a 562-bed tertiary academic medical center. We are a regional referral center for approximately 1 million people in Vermont and northern New York. And we're part of the University of Vermont network, which consists of 6 hospitals, a home health hospice agency, and many medical groups serving patients in Vermont and northern New York. In addition to that, we also have something where we have something called NECLA, which is the Northeast Community Alliance, which is our laboratory is part of an alliance with all the hospitals throughout the state of Vermont, as well as New York, Upper New York State. And we provide reference testing for them, and what we cannot provide, we send them on to Mayo Medical Center. So every day we have quite a few specimens coming in. Prior to October 2018, when we went live with Fusion, we were using the Prodessa assays, the ProFlu, the ParaFlu, the Meta Adeno. In the past, we did use ProFast, but we had stopped using it several years prior. And we also have GeneXpert Express Flu RSV for our stat samples, ED, and inpatients. The Prodessa assays, we use the EasyMag instruments for extraction, and we use the Smart Cyclers for amplification. In the past, prior to 2018, the ordering practices for physicians, they could order this pretty much a la carte. They could order it under one viral detection order. They could pick and choose whatever they wanted. The majority of the testing prior to 2018, October 2018, was focused to meet the needs of our flu RSV testing and with some respiratory virus testing to be performed for inpatient oncology patients in other Vermont hospitals. We ran flu RSV on GeneXpert Express, like I said before, for our emergency department and for inpatients to provide that quick turnaround time that they require, and I'll get into why in a little bit. The Prodessa assays, when we were running them, we were doing them in batches, Monday through Friday, outside of the respiratory season. And during the season, we were trying to do them 6 to 7 days a week during the actual season. We typically ran 1 batch a day, but there were several days where we had to do 2 batches due to the volume. Our average volume, as you can see here for the various assays, as well as we had 2 current Panthers, and we used them for CTGC and HPV testing. And in December of last year, we did bring on HPV genotyping as well. So why did we decide to make a change? Well, Cepheid had told us as of December of 2018, they would no longer support the Smart Cycler instruments or its consumables. So that was a big reason for us to go. But there was also another really big reason, as we started seeing many, many unresolved indeterminate, what we call indeterminate results with the Prodessa assay, since we switched to using a flock swab and M6 media as our collection method. We decided as a whole microbiology lab to switch to the flock swabs. We use the e-swab for bacteriology in anticipation of hopefully in the next year or two going to automation for microbiology. And so we switched for virology as well. And we started seeing problems almost immediately when we switched. And so we did a lot of repeat testing, and you'll see I'm going to get into that because they would come out unresolved. And upon repeat a second time, if they were also unresolved, we called them indeterminate, which started causing a lot of problems with patient management and obviously having to repeat many tests, decreasing our efficiency and increasing our costs. And more important, and another important thing is frustrating the lab staff who has to repeat all this testing. So back in 2017, I attended CVS and I started to look at the newer platforms to start our decision matrix for our lab and figure out what we wanted to do. I looked at both syndromic assays and modular assays. We were looking to improve our turnaround time, make it simple to use. The cost was important. Instrumentation footprint was important, and to provide appropriate testing for our patient population, and also finding a really good test. We narrowed down our search to one syndromic assay. We decided the GenMark ePlex RP assay and the Panther Fusion module. We brought in the GenMark and the Panther Fusion. We put a fusion on one of our Panthers and we brought in the GenMark for a trial. And we really wanted to throw a bunch of specimens at these instruments and see how they really performed with our samples. And we really wanted to see, we saved a lot of our unresolved samples to see how it worked for those samples. And I will be talking about that in a little bit. So in our initial testing of these systems, we saw comparable results with both platforms. Those previous unresolved and determinate samples on each platform was not an issue on these newer platforms. They worked great. And we did find several positives that were in that group of unresolved. Prior to initiating a full verification, we decided we needed to really start looking at how we wanted to go as a laboratory and for our institution, whether we wanted to go along the lines of having more modular type assays or a syndromic assay. And we really had to step back and look at our patient population. And we also had to conserve our bank of frozen samples. We don't have a huge, we started saving for this project, but we don't have a large storage space to keep a lot of samples. And also the cost of verifying both assays was a factor as well. So we looked at that. So based on our patient population, we do not have a large transplant center at our institution. So we decided that the Panther Fusion modular assay best met the needs of our laboratory rather than a larger syndromic panel. Both platforms did perform well. They were both easy to use. The space requirements for the Fusion were a little larger, but the ability to add the Fusion module onto our existing Panthers, the space that we had where our Panthers were accommodating to those Panther Fusion modules without a problem. And during downtime, GenMark ePlex does have redundancy in their modules, and they have multiple modules, so if one's down, the other one will be up. But we did see in our trial period some downtime with modules in the ePlex. And the assay costs were higher for ePlex. So we went ahead and did our validation, fuller validation on the Panther Fusion, and this is our MP swabs. And you'll see here that it performed very well with the MP swabs. Obviously, we didn't have para-4 because we had never tested for para-4 in the past or rhinovirus, but we did find in some of our negatives that we did have rhinovirus, which we expected to find. We did keep the GenMark around because we had to be able to test samples that were positive for rhinovirus or para4 on another platform. So we did keep it around for that to help us out with that. So in our verification, we had one positive sample for adeno that was positive by Panther Fusion with a CT value of 35.2, but was negative by the Prodessa assay. And there was insufficient sample remaining for additional testing. And one sample was positive by Panther Fusion for rhinovirus, but it was negative by the GenMark Eplex assay as well. But we definitely see greater than 95% comparability here in our accuracy. We also went on because we are a reference lab, and as well as we get inpatients where we want to do respiratory fluids. So we decided as a modified assay to look at respiratory fluids, and we did a bigger validation for that. And we saw only one discordant sample for para2 where a sample was spiked into a BAL specimen that was tested on fusion was negative. And so my question was, because we had other ones spiked at the same time by different techs that worked fine, the question I had in my mind was this spiked correctly? But still we saw greater than 95% or close to 100% on almost everything. So we started to think, okay, we're done our verification. How are we going to roll this out and how are we going to use this for our ordering practices? So we decided in a meeting with many players in the room to decide to do the emergency room and the urgent care center to continue to use GeneXpert. And so we created a separate order code just for GeneXpert because this test is good for turnaround time, but it also costs more. And so our marketing people as well said that we really need to create a separate code for that. Inpatient and outpatient flu RSV testing we were going to do on the Panther Fusion. Outpatient and inpatients have a separate order code, so when they come in, they go directly to the Panther Fusion. Then we decided to combine the para-influenza and the adeno-meta-rhino assay into what we call an expanded respiratory panel, where they order that and they get both regardless. We were not going to do it as an a la carte because we felt that that respiratory panel was really meant for severely ill or immunocompromised patients, and if they are severely ill and immunocompromised, they should be doing both. So soon after we started, I will get into this. Currently running the Panther Fusion assays, we are running them 7 days a week now as on demand. This improved turnaround time now allows for more timely patient results and more effective patient management, including it was well received by our infection control team. They were very excited to see this. It's our long-term goal to add this testing to our evening shift. When we complete that automation project and do some spatial planning, we will be able to move our Panther Fusions in a more conducive area where it's closer to our evening shift where they work in the receiving area. Soon after we started into the season, probably about November or December, we started to see some issues that the hospital was having. Our hospital currently has very few single bed rooms. And so not getting flu results for inpatients in less than 30 minutes on the Fusion did cause some patient placement issues. So subsequently, we decided to perform the inpatients on the GeneXpert to provide that turnaround time. But our hospital is currently going to be opening a new single bed tower starting next month, and they're going to retrofit a lot of the other rooms to be single beds. So we will definitely reevaluate once that opens up whether we need to continue that, because we would prefer to go back and do inpatients as on the Fusion. So I also wanted to look at our repeat testing and see have we improved? Because that's one of the reasons we made this change. And so I went back and looked in the same time period. So I picked October 25th of 2017 through March 31st, and that's where we were running Prodessa. And then I picked October 25th through March 31st on the Fusion. So as you can see, I'm going to go through the different assays. So for ProFlu, we were seeing a high number of repeats at 19.3%. All those repeats required repeat testing and sometimes 24 or 48 hours to get that done, at a cost of over $9,000 to us to repeat all that testing. When we went to Fusion, we saw 2.8%, and repeat testing took less than 3 hours because you can put it right back on at a cost of about $1,300. Then we also looked at our issue with Adno and Meta, and Adno we didn't have a big problem with unresolved at only 0.8%, but Meta was a big problem, and it was our biggest problem. We saw 36.7% of our Meta tests had to be repeated, again, creating more time to get resolved at a cost of over $12,000 to us. We did see 6.5% repeat testing. I will say some of that percentage is due to the fact the instrument went down and we lost some testing during this time period. But I'll get into downtime because I know I always ask that in a few minutes. And again, that repeat testing was only $1,300. We also looked at Paraflu. Again, we had a high number for Paraflu at 21.3% with Prodessa at a cost of over $4,300. And with Fusion, it was down to 3.3% at a cost of only $700. So overall, we looked at our cost savings because up above, and they always want to look at the hospital management. Are we saving money when we bring in this instrument? Because these instruments, everything costs money and they want to know we're improving. So our Prodessa assay repeat testing cost us $25,991 during that time period. And for the Fusion during the same time period, it was only $3,400. And that's a savings to us of over $22,000. So testing on the Panther Fusion, a single extraction can be used to test for all 3 Panther Fusion assays. They take roughly 2.4 hours for the initial result, then 5 results every 5 minutes. It's a random access. You can do 335 results in 8 hours on the Fusion side. And if you add in the Aptima side, you get up to 500 tests in 8 hours. And it's the only IVD assay where enterovirus and rhinovirus do not cross-react. It does have the ability on the Fusion to run TMA, LDTs, and open access, which I think Kathy's going to talk about a little bit, and PCR assays simultaneously. And the assay-specific reagents are in cassettes, which our techs love, no preparation. They're just pop them on. There are 12 tests in each cassette, and they can stay on board for 60 days. We're currently using the Panthers for CTGC HPV, and we added genotyping in December. And previously, training on the Panther for previous analysts only took about an hour to teach them how to do Fusion. So the trained Panther users, it was very easy to learn. And probably for me, it took about 20 minutes. So new Panther analysts, we still allow for 1 week of training, and that's more to get used to our workflows in our laboratory. It's not that hard to train. This is very user-friendly. So based on this, we used to, in the past, have 2 virologists that performed the respiratory assays, and HSV, VZV, and CMV Monday through Friday, and 1 virologist on the weekend. Now we're currently able to absorb those viral respiratory assays with our current Panther bench duty. So our Panther person does Fusion and all the Aptima assays, as well as some days they are doing, we have a Tomcat that does the aliquoting of HPV. They're involved in that. So they're doing a lot. They're busy, but they can do it in one day and usually without an issue. Our HSV and VZV, we brought in Luminex Aries, and we decided to combine a bench that we had a BD Max, we have the Gene Expert, and we decided to put Luminex all on one molecular bench. So basically that freed up one person that we had. And so we used that savings of that technologist to use to balance out workload that we were seeing in some of our bacteriology areas as well as our receiving area. So we use that technologist so we have a savings of technologists. So everybody always asks, and I ask, downtime, what is your downtime or what have you seen? Well, because we have 2 Panther Fusions, and with that redundancy, our lab was able to provide consistent laboratory service during the respiratory viral season, even if one instrument was down. With each respective instrument, if the Panther Fusion module goes down, it does not stop the Panther Aptima side, and I have seen it, it does not. Within each respective instrument, if the Panther Aptima assay side goes down, then both sides are down. So I wanted to show you really what we saw during our experience, and we did have some downtime we call them Pan 1 and Pan 2. We did see some downtime, but it's usually less than 24 hours. We give them our logs, they order the parts, the engineer comes on site, and it's done. We did have one Panther that was down a little longer because they discovered something else that they had to get a part for. But we were never ever down because we had to. So in conclusion, I would like to say the Panther Fusion assay has improved our results, eliminated our issue of unresolved or indeterminate results. This significantly improved patient management and infection control practices. They love us now. And the new viral assay has improved efficiency in our laboratory, having us transition from a very labor-intensive assay to automated methods. And I can't say this enough, the technologist's job satisfaction has been noted due to this simplicity of the test and less repeat testing. And with an IQCP in place, which we have on our Fusion side, we were able to reduce the amount of QC we do for each assay to new lots, new shipments, and we chose every 30 days because all of Micro tends to do ours to keep it simple for the techs to remember 30 days for all our IQCPs. And so based on that, we saw decreased costs and less repeat testing and less QC testing with the newer platform. And I didn't add it on here, but I would like to say that I do appreciate, I've been working with Hologic for a long time, and I do appreciate their great service. I do appreciate their ability to listen to you when you have suggestions, and I do appreciate that they try to be innovative and come up with solutions to issues that their users do bring to the table. So I do appreciate that. And if you have time, go see their booth because they are even improving some of this with new add-ons that they're going to be doing. So I'm excited.